back for day 2. Thanks for joining us again. Good morning to the East Coast, very early morning to the West Coast. Happy afternoon to the European folks joining us. Thanks. I just wanted to real briefly introduce what we're going to discuss today. Hopefully we'll have some presentations in the first session and then more discussions in the afternoon. The topic for today, topics, because they're a bit disparate, are interlaboratory comparisons. And then we also want to address definitions and practices for reporting uncertainties. Before I get into that, I wanted to just real quickly, very briefly, give a summary of what we talked about yesterday. So the main theme yesterday was methods, as you know. The topics kind of all fell into a few different categories: collection, preservation, and storage of seawater samples for DIC isotopes. We discussed various different extraction methods, the different isotope analyses for C13 and C14, and standards were briefly addressed by several different groups, the types of standards they're using in their labs. For the collection, preservation, and storage, there was some debate or discussion about glass bottles versus the foil bags. It seems that mercuric chloride poisoning is still sort of the gold standard, but there's still obviously ongoing issues with its toxicity and difficulty getting it around the world and on ships. So freezing and copper sulfate were discussed as other preservation methods. And then storage time, as many of these samples stick around in one's lab before we're able to process a whole cruise worth repeatedly. So the discussion of how long we can keep these samples around and the stability of the storage methods that we're using. For the various extraction methods, I think there were basically 3 methods: the sparging technique, headspace extraction, and then the membrane transfer, the Replicas system. For the isotope analyses, obviously anyone doing C14 on DIC and seawater is using AMS, but we had some presentations and some discussions about using IRMS, both dual inlet and continuous flow systems for C13, as well as a nice presentation and discussion about using cavity ring down spectrometry. And then the standards, this is not an exhaustive list and it's a bit of a mismatch, but I think that's kind of what it felt like yesterday. Everyone's using a bit of a mismatch of different standards. There's certainly certified standards for C13 and C14 that are in use in different labs in various different ways. Everyone's trying to get a good handle on their own internal standards. Some labs are using dissolved carbonates or sodium bicarbonate powder. Other groups have an internal seawater or artificial seawater standards. Certainly people using IRMS systems and cavity ring-down systems have a CO2 gas reference material in lab. And there was many mentions of the Dixon reference material, which will come up, if not today, then certainly in some detail tomorrow. So just wanted to sum all that up. I apologize if I missed your favorite standard or your favorite storage technique, but I think I covered the highlights there. And all the details are still available in that Google Doc that the different groups, the discussion groups put together yesterday. For the agenda today, this will just be really brief. For the morning session, we're going to have a couple presentations. Anne and Maxi are going to present some historical interlaboratory comparisons. So Anne will go over kind of what has existed in the past as the seawater isotopes program was being created at NOSAMS and as part of the WHOI and Kleiber and Goship programs. And Maxi has a really nice comparison, very recent comparison between NOSAMS and ETH for seawater C14. So we'll have a look at those. After that, we're going to switch gears a little bit and start talking about data. Bob is going to give us a presentation to talk about database level data processing and reporting. And then I hope that will lead into our discussions this afternoon, where we can talk about future interlaboratory comparisons, how we might go about doing that as a group, but then also from the data level, how each individual lab is processing their data and reporting uncertainties so that we can achieve a level where everyone's data can be used by the public and get into these databases. And then we'll sum up at the end of the day. So I just wanted to, that's all I have. I just wanted to give a brief overview. If anyone has any comments about yesterday that they want to bring up now, we can do that now. Otherwise, we can go ahead and lead into Anne.
Do you see that comment?
Yeah, I hope Bob will talk a little bit about some of the global databases for seawater DIC, which many of you are oceanographic data in general, I guess. But yeah, it would be great if we could address a centralized database for these types of measurements, maybe within this group or sort of a larger part of the field. So keep that in mind. That's great. Amanda, thanks.
So shall I take over?
Yeah, sorry, Anne, if nobody has anything. We're still waking up here, I guess.
Oh, I do have. What is going wrong here?
Your far left, play from start.
Didn't do that yesterday. Thank you.
No worries.
So this is, I'm using a lot of my slides from a previous presentation, but basically, as those of us who are here know, both DIC13 and DIC14 are powerful tracers of the carbon cycle and ocean circulation. NOSAMS has contributed over 30,000 measurements to the global databases. This in spite of there being no recognized reference material. The measurements are generally comparable, but would be better with a certified reference material. I think that Bob Key will be addressing that a little bit in his talk later this morning, perhaps. As we heard yesterday, the current practices are to use IAEA and NIST isotope standards, as well as laboratory produced secondary standards. And for larger comparisons, there's deep water comparisons and interlaboratory comparisons. So with respect to carbon 14, early on in NOSAMS history, as the WHOI program was gearing up, the only way we could ensure that the radiocarbon results were comparable was to look at deep water in the Pacific. And so the stations listed here are shown on this map of the Pacific. So most of the results came from the Northeast Pacific. And stations 201 and 202 were collected by Ostland's and measured in Ostland's lab as part of the GeoSex program in 1973. And station 12 was occupied in 1991. And what we're looking for in comparing these is the difference at the surface. But the deep water, there hasn't been enough time for the radiocarbon to change, and we can see that there is good agreement between the two profiles, which is what one would hope to find. And then for a direct comparison of the large volume method and the AMS method, so counting versus AMS, during a cruise at Station 48, samples were collected by Ostland's group and for NOSAMS. And we can see that there is, again, very good agreement between the two methods. So this gave, although it's not a lot of data for comparison, it was allowed us to be comfortable that we were doing a comparable job to the large volume sampling that had been done up until this program started. The concept of how long can you store a sample was also right at the forefront of NOSAMS, because samples were being collected before the accelerator had been delivered to the laboratory. And so during the cruise P6C, which is along 30 degrees south in the Pacific Ocean, one station, 48 samples were collected. It was also a GeoSex reoccupation site. And were analyzed in replicate sets over 5 years from the date of collection. And you can see that the value you expect for 2450 meters from the GeoSex results are that everything we measured except for one pair agrees very well. And it's, if you include even the pair at approximately 500 days, there's a plus or minus 8 per mil. If you exclude that, I actually don't have my notes showing up. It's less than 8 per mil. So this is actually very encouraging. So these samples were collected in 500 milliliter bottles with ground glass stoppers using APAs on M grease, I believe, in a rubber band. One thing that wasn't mentioned yesterday was the bottles that have been used for the WHOI program were specifically used a very high quality ground glass stopper. And I don't know if that's something that needs to be thought of as bottles go forward. So this is actually, I think, a really nice piece of data set. And also, now I'm moving seamlessly to Delta C13. And here we do a lot of comparisons with the UW lab. But this has happened to be a case where UW and NOSAMS were on cruises from different programs, but they happened to be at very similar sites. And you can see that they were collected within months of each other, and that there is good agreement between the UW and NOSAMS measurements, with the exception of a point or two. So that's gave us confidence again. The only really organized Delta C13 comparison that I'm aware of happened in 2001. And I'm embarrassed to say I can't remember which laboratory organized it. It wasn't NOSAMS, and I don't think it was UW, but there were 1, 2, 3, 4, 5, 6 labs participating. We were given tubes of gas as well as a bottles of water. And you can see that there's much better agreement in the gas replicates than in the water stripping replicates, which is not that surprising. There's a number of labs that are all very similar in the water stripping, and then a couple of us that are out here. This doesn't mean the data are bad. It just emphasizes the point that having a standard to calibrate to would make the results that we get going forward much better. And finally, I wanted to touch on the Chang et al. Lab comparison experiment that Kalina talked about. Basically, Doug Wallace's lab at Dalhousie organized a suite of ocean water samples designated DSW, deep sea water, and a batch of Andrew Dixon's reference material to be sent out to all the participating labs. I'm going to mention this. Apparently, the notification for this workshop went out on one listserv that nobody at NOSAMS happened to be a part of. So I just want to point that out as when you're doing comparisons of this sort, it's useful to use more than one venue to advertise experiments like this. But it's a great experiment. And as Kalina showed you, we were able to measure the reference material within the bounds of what everyone else measured. So the top graph is originally just what everyone reported for the reference material and for the deep sea water. And there's a fair amount of variability. The GOOS program, the Global Ocean Observing System, has identified C13 as I don't know what they call it, but a major variable. And it has stated that you need plus or minus 0.05 per mil reproducibility in your measurement for these to be useful in that program. Clearly up here, you're not getting it. But if you assume a certain value, a stated value for the reference material, and everyone calibrates their results to that value, they get a much tighter correlation for the deep sea water. So the results of all the laboratories was about plus or minus 0.06 per mil, which is very close to what is required. So that was an excellent comparison study, and it would be good to do that for carbon-14 as well as C13 again. So there's a need for standard isotopic methods, and I don't mean everyone has to do one stripping method, but just a way to establish the equivalency of the methods, reference materials, and procedures for reporting uncertainties, which Bob will talk about more later. So that's it. Thank you. I think there's essential ocean variable. Rick Wanikoff has said. Okay, thank you.
And there was one other thing we did in WHOI started in the Pacific, and for the entire Pacific operation, we used AMS samples for the upper more or less 1,500 meters, and we used large volume samples for deep water. Now, there are a couple of cruises that are exceptions to that rule, but the idea was to give NOSAMS time to get the kinks worked out of the procedure and to prove to the community that the AMS technique was good. We had 20 years of experience or more. I guess we had 40 years of experience with the large volume techniques. So on, I would say something like 75% of the cruises, we overlap the AMS and the large volume samples at depths around 1,500 meters. And I guess I was the only one. We never published that comparison other than sort of in passing when the various legs were written up. We would mention how well those compared and so forth, but there's never been an overall synthesis of that. But the data looked quite similar to what Ann showed in the various plots there up in the northeastern Pacific. That was supposed to, or at least we had planned on continuing that into the Indian Ocean WHOI program, which was done next. But on P10, the leg that it's the westernmost leg that the U.S. participated in, we were about 2/3 of the way through the cruise, and the guy running the winch fell asleep at the winch and pulled one of the Girard barrels into the block, broke the cable. We lost 7 out of the 10 Girard barrels we had on board, and that sort of ended large volume C14 for all time. And in spite of getting to be very, very good at hanging those monstrous barrels, I was not too unhappy to see them disappear.
Wow, Bob, thanks. That's quite a nice story, maybe.
Yeah, well, we trolled for them for about 4 hours, and
wow.
I would never have bothered to waste the ship time, but the people on board assured me that they had had quite good luck trolling for when you lost that much wire. But anyway, with 7 Girard barrels and something like 3,000 meters of wire on the seafloor, we still couldn't hook anything. So they're down there. I can tell you exactly where they are if you'd like to go troll for stainless steel Girard barrels.
Well, it's great that we managed to make measurements on much smaller volumes of seawater. Then that worked out well.
Yes, we roughly a factor of 1,000. Those were 50-gallon samples in the Girard barrels. And just for the scientific aspects of this, if you want to look at 1990s era C14 Pacific results and you go to any of the databases and pick up the raw data, then you need to get two files because the large volume data and the regular rosette data are stored in separate files. The units and the columns may not be a one-to-one matchup, but it's pretty easy to put them together. And the alternative to that is to get GLODAP where they already have been merged. C13 data out of the Gerard barrels is garbage. Well, scientifically, it was, it's okay to, its only real purpose was to be there to correct for, now, help me out, Roberta.
Delta C14 calculation requires a C13 correction so that they're all
for the
at minus 25 per mil. So.
Right.
So
that's historically true.
Yeah, but it's still true. So
there's one
exception. Don't need the external measurement anymore. So.
Yeah, we can just do the correction online.
That's right. The only group that ever successfully got reasonable quality C13 measurements out of that technique was Wolfgang Reuter's lab in Germany when they were in Heidelberg primarily. But the US C13 measurements, something about the extraction equipment made the C13s they had too big errors in them. And that goes all the way back prior to geosecs.
Well, it's good that we managed to improve on that too. Sorry, Anne, I just wanted to bring it back in case anyone had any comments about the interlab comparisons.
Yeah, so I actually have a comment. I have this feeling that there are more interlab comparisons out there, but they're not all organized or published, and might be useful to. I don't know how we could figure out a way to get them all together, but.
Yeah, and I'm sure there are people at this meeting, and noted that NOSAMS wasn't part of the 2019 paper, but I'm sure some of you here were. So if you want to chime in about that process, or we'll have time to discuss what we want to do in the future, but now's a good time to comment on sort of the best ways to organize these things and what we need to have a good comparison.
I have a quick question for you, Anne.
Mm-hmm.
In your presentation, the Cheng et al. Interlab comparison, the upper plot is the raw results.
Yes.
For C13. And what did they do in the lower plot? They normalized to an agreed upon value. Is that what's happening?
Yes.
Okay, so this is where Doug Wallace or whoever advertised what the accepted value is, and people normalized to that.
Yes, and my understanding is, I assume he just took what he measured and said, okay, this is what it is.
Okay, so we don't know the value, we just have an agreed upon
Or
value.
I think, did they average everybody's and then say?
Yeah, average everybody, and they use the average.
Yeah, okay. My bad, sorry.
Okay, that's it, thanks.
Yep.
We have a couple of minutes, but we can also go to the next comparison. But just if anyone has any comments about that study or any of the more historical ones, going back to Bob's loss of large volume samplers.
We can't. I assume you wanted to be muted, Masha. Mashi.
Hi. So, sorry. So as I told you, I am on holidays in the Canary Islands. Somehow they decided to do maintenance work in the building, and the electricity is gone. No Wi-Fi, so I'm using my the data from my phone. So stop my video, hoping that it will work. All right.
But
I can share your slides.
Yeah, we can find a, yeah.
I mean, can you take the last version that is on the drive?
Yeah, I'll grab the most recent version right now, and then I'll share as soon as I have it.
Yeah, and we'll go ahead and get that going, Mashi, so you can get to the other side of this.
It was totally unlucky. Like 5 minutes ago, I was like, no, this is not possible. Right.
Yes, it is.
Where is that picture, Anne?
That is at the Rockefeller University in Manhattan. I was synthesizing peptides in Robert Merrifield's lab. I always like to say he won the Nobel Prize. He won the Nobel Prize 2 years after I left the lab.
Clearly, it was your work.
Yeah.
I have a comment. I don't know if you can hear well with my microphone. I don't know if this is the appropriate time to bring it up, but 2 standard normalization is super important to account for variability and comparability. You know, so I know that it's easy to come up with a standard that's close to 0, but having a very light standard to fully bracket expected values is super, super important.
Yeah, I agree. Yeah. Thanks, Lydia.
The USGS 44, I think, is important, even if you're not measuring samples that are that light. If we, agreeing on what the scale is.
Yeah, I agree. That's a really good point. Let's go ahead and let Mashi get this presentation going while he still has a little bit of connectivity, and then we'll talk about the data for sure. And I think that's an important point, so we can bring that up again afterwards. Thank you.
I'll try to be brief, and if you cannot hear me very well, just let me know, right? So we got our data from the ocean. Then I did compare our data to deep water data from previous cruises and so on, and we were quite confident that the data were looking good and that we were doing a good job. But of course, you always want to compare your results to a laboratory that has very long time experience. The intercomparison of DI C14 in seawater between the 6 TTH and NOSAMS. May, if you can pass to next. Yeah, so we were lucky to start the collaboration with Heather Graben because her group took, well, Joe Lester collected samples along this section shown in the green line in the globe there, across the Florida Strait and the Canaries between 23 and 28 North. The cruise took place in winter 2020, and Joe Lester collected 15 replicates. One set of samples was collected in glass flasks that were collected exactly as stated in the guidance report, Collection and Measurement of Carbon Isotopes in Seawater DIC by McNichol. And the other 15 were collected in the foil bags and preserved by freezing. One of the bags broke, so we can only compare 14 of these replicates. The results of the section, you can see them here. And the stars, they show where the samples were taken. So they were taken so that they would cover surface to very deep waters and different parts of the Atlantic. And as you can see, they basically cover the whole range of DIC, delta C14, spanning from -130 to about 60 or so. If you can move it to the next slide. Perfect. So about a year later, we got these foil bags from Heather Graben, and we stored them in our freezers. And we started analysis about 14 months after the sample collection, so in April 2021. So as I told yesterday, we would let them unfreeze overnight, transfer them the day after to our glass bottles, and then we would freeze straight away the samples. So in the next slide, I'm just gonna show you again, for those that were not attending, this is the method that we use. It's a single line coupling both the extraction and graphitization steps, and it's all fully automated. And here you can see the reference to the paper if you wanna know more about it. And now I'm gonna show you the results in the next slide. So we have 14 replicates. The yellow bars, they represent the results reported by NOSAMS. The green ones represent the results from the Laboratory of Ion Beam Physics. And we were very happy to see that the majority of the individual measurements, they agreed within uncertainties. Nonetheless, there seems to be a small offset. ETH, or our laboratory, seems to be providing slightly lower results with a mean value of minus 1.4 per mil, which is, well, it's an offset that we would probably need to do more replicates in order to more accurately determine how, well, to accurately determine the offset, but we are in any case very happy about this. Just to conclude, again, I wanted to acknowledge in the next slide to Joe Lester, Heather Graven, and the fantastic team at LIP. That's it. So yeah, I thought it was better to be brief than to extend too much in this, but we are very happy, I have to say, and perhaps we would need to discuss whether we need to do more replicates or not. And my questions would be more regarding in the future, whether how often we should do intercomparisons. Is it something we should do for every cruise? Is it doable or not? And so on. So probably this is something we can discuss in the breakout groups.
Yeah, thanks, Maxi. I have to say, those of us at NOSAMS were also very pleased to see those results. It's always good to know that that we're all making as high quality measurements as we think we can. Does anyone have any questions about the intercomparison? I see in the chat, Polina asked where you purchased the bags from, although, and what the cost. I know you don't buy them, Maxi, so maybe, I don't know if
yeah,
Heather
but Heather is around, so maybe she can say something about this. I'm gonna check the chat.
Yeah, I can read these out. There's a question about any hypothesis about the small offset. I don't know if you have any thoughts. For my first thing, I think we should go look at maybe some of the other types of standard, not even seawater, other things that we measure between the two groups. 1.4 is I think outstanding. It's certainly within our measurement error, but maybe you have some thoughts about why it would be consistently lower. I think you're on mute, Maxi, I'm sorry.
So Rick, I haven't thought about this in detail because truth is we got the intercomparison results the week I was leaving ETH. And so I don't know why of this, what can create this small offset at this point.
And as Roberta said, that is amazing in comparison. So you know, it's probably very close to the experimental error, but you are showing it's just statistically meaningful just because you have such excellent results and precision.
So thanks a lot. So I mean, we were very happy. But I mean, the thing is, I don't know how is it done. So we are new on this, not in C14, not in AMS, not in marine work, but we are new in doing DIC14 in seawater. And so I'm not aware whether in the past you were doing more replicates or not, or whether you consider 14 samples it's enough. I mean, for us, it's pretty good to know that we have already small offset, and we know how much of offset we have, and that the offset is usually let's say, smaller than what you can find in the uncertainties reported by different laboratories. So it's very good. But I don't know what you used to do in the past, whether you were doing more replicates or not, or.
So that I think is the point of this workshop, is to help us figure out how frequently this kind of stuff should be done and how often it needs to be done. It needs to be done more frequently if there is no agreed upon standard for everyone to use. And as part of the WHOS program, WHOS Clive we arranged to have duplicate samples collected on every cruise. So we have what I call shipboard replicates that give us a measure of how reproducible the process is from ship to shore. So duplicates are always a good thing in chemical measurements. So, I mean, it's fairly clear that what will come out of this workshop is a need for reference materials and more comparison experiments. So.
Yeah, one thing that intrigued me as well is that the sampling method was quite different, and you actually did a transfer from a unpoisoned bag. And we heard earlier on that there is a lot of concern in the community about the ability to use preservatives, for carbon isotopes, but also for the total inorganic carbon. So there were several steps, and I think in that respect, C14 is a little more forgiving, because you can, you do a first order correction, fractionation correction using C13. And there was a question as well about if the C13 was measured on these samples. So is that, I'd be curious to hear the answer to that as well.
No, we didn't measure the C13 itself. We used the C13 just for correction of the fractionation, but we don't have the proper C13 data. Not enough accurate, let's say.
Brett Walker had a comment asking, or would you like to unmute yourself and ask it, Brett? Or
yeah, I see.
Sure.
Freezing seawater often results in precipitation of carbon-bearing salts that take a while to get back into solution at pH 1-2. You have 100% yield of DIC with this approach. Perhaps this could contribute to some C14 upstream. No, we don't have 100% yield of DIC with this approach. So perhaps this is one reason. I have to show the expert on this. I'm pretty new here. The expert on this is Lucas Wacker, and I'll discuss this point with him to know what he thinks about it. I'm gonna note it down.
Just a thought. We don't always see it when we freeze seawater samples. It seems to be intermittent and sometimes based on the environment you sample, but something to look out for.
Okay. Then, how do blanks look like with your method? The blanks look good. So yesterday I mentioned we do two types of blanks. One blank is a procedural blank that it's now getting close to the combustion blank. The combustion blank is the phthalic acid that it's combusting directly into the elemental analyzer. And the procedural blank is meant to represent the extraction and graphitization steps. And they look, the value of them is for the combustion blank is 0.002 fraction modern. And the procedural blank, it's at present is about 10%, 20% larger.
Thanks, Brett. I don't know if you have a follow-up on that. Thanks everyone for the discussion here. I think we can go ahead and switch over to Bob. Thanks, Mashi, for getting that in while you had no power or internet. That's really nice, but hopefully you can stick around for the discussion so that we can get back.
Yeah, hopefully Wi-Fi will be back.
Okay,
Sure. Thanks.
great, thank you. Yeah, and Bob, do you want to go ahead and oh, Heather, do you have something? Do you want to transition in?
I could just mention
Thank-
before you switch. So we collected samples using the same method with the bags and freezing on another cruise 2 years earlier. And those we measured at the NERC radiocarbon facility in East Kilbride in Scotland. And we also did a comparison with NOSAMS. And that intercomparison wasn't as good as Maxi is showing here. So either there was some problems with the sampling or the lab in Scotland, the measurements aren't as good as Maxi is giving in ETH. So we did have an offset of about 6 per mil between the two labs based on a few more samples, about 30 intercomparison samples. And we also measured C13 in the East Kilbride lab there in the bags, and there were quite a few outliers and a lot of scatter. So we've kind of not really looked at that data, presuming it's not that great, but we haven't had a lot of time to really go into that and examine what's happened there. So just to mention those few things. And then one other thing I wanted to mention, which is a bit unrelated, is just that we have a couple of papers where we've done model simulations projecting C13 and C14 in the ocean under different scenarios. And the model output is available from those papers. So in case people weren't aware of those and those could be useful, I'll put the links to the papers in the chat. Thanks.
That's great. Thanks, Heather. Yeah, I think it's what's most encouraging about this most recent study is the 14 months, that the fact that they were able to store that samples for that long as well in this method and get such comparable results. So anyways, thanks. I'll let Bob take over. And,
Okay. Yeah, that's some of the issues that Heather was talking about are some of the inherent problems with laboratory comparison studies. When you get a different, if things are as good as 1 per mil as they were in Mashi's test, then you go out and you have a cold beer and celebrate. On the other hand, when things don't go well, all you know is that they're different. And you've got myriad possibilities with real samples on where that difference might be. And it sends you off on a whole scavenger hunt trying to find out where the differences are. If, on the other hand, you have a certified reference material, then most of those issues go away and sort of you give up the quest for absolute accuracy in exchange for producing comparable data sets. Because all you're trying to do is reproduce the reference material value as closely as you can. But anyway, it's just sort of a different philosophy. All right, I'm going to talk about what happens after the numbers are produced. This is a procedure that I developed with NOSAMS over the years as they were learning how to make measurements, and I was learning how to deal with WHOIS format files and build large data sets and so forth. In no way do I mean to imply that this is the only way to do it, or even that it is the best way to do it. It's just the way that we have done it. So it's good for everyone to know what it is, simply because we've put so many numbers out on the in the public realm using these techniques, and the techniques have been I'm pretty sure completely consistent since the first WHOIS cruises, which were up in the Northeastern Pacific in 1991. So we're going to look at how the data gets from the lab to the data centers. Kathy Elder at, you can see this is just literally a snapshot of a email that I got from Kathy this year, last year, 2020. The data is from P18, and there are two things in this message. One, she tells me what the data are from. This particular number is called an expo code, It is a unique number generated for every major cruise that goes out at sea. I expect most of you are familiar with these numbers, but in case you're not, 33 is an index for the country. In this case, 30, I think it's 32, 33, 34, the US. RO is a code for the ship. In this case, it's the Ron Brown. And then the rest of that is just the date that the ship left port. So November the 19th of 2016, the ship sailed from port. And P18 is the north-south section in the eastern Pacific that terminates on the north end, I think, into Mexico. So Kathy sent me this after they had finished making all of the measurements, and in this case, she is pointing out a subset of the samples listed that, in the opinion of the lab, had questionable results. Now, you'll see in the next slide that she does not provide flags to me. The only other important thing to note from this, and I would recommend that everyone adopt this if you're not already, and that is the identification system that's been used for decades, and it works beautifully. It's also guaranteed unique. And that is simple combination of station, CAS, and the Niskin bottle. Now, sometimes the Niskin bottle can get to be unwieldy. It could be a 3 or 4 digit number, and that just gets to be error prone trying to write it down. So on Ghost Ships cruises, in addition to the Niskin number, we also give a sample number, which is just 1 through N, where N is the number of Niskin bottles on the rosette. Anyway, so this is a note that, that Kathy sent me. And at the end of it, she says also that here's a summary of the C14 duplicates for this particular cruise. 13 pairs of shipboard replicates had a standard deviation of 2.6 per mil. The file that she sent, this is a subset of that file. Exactly, again, it's just a cut and paste. I don't know where the red dots came from, just ignore those. The cruise, on this particular case, we had 543 total analyses. That includes duplicates. And again, here to begin with, we've got the ID information. This is a unique number that we have etched into each one of our C14 bottles. The C14 results from the AMS is produced by the lab. Kathy reports 2 errors to me, an internal and an external error. The internal error is mainly just a combination of the various counting errors that come up, the blank and the literally the number of atoms counted. The external error can be larger or smaller. Typically, if it's different from the internal error, it will be larger, and it would be the results, a standard deviation of multiple analyses of the same target, so the same graphite target. This OS number is a number that's specific to Woods Hole. Well, we don't keep that for the final records that go into the various data centers, but it's something you have to have. At least we have to have it, because it makes life easier if I want to go back to NOSAMS and say, could you track down all the information you have on a given sample? It's the number they use to reference things internally. And then that last number, which for me is underneath all of everyone's picture, is the C13 result. So anyway, this would have a whole bunch of results. In this particular one from CAS Station 1 and these CAS, there are no replicates, but we've got one sample down here where something went wrong. Doesn't say what went wrong, but there are no results for C14 or C13. So this sample would be initialized with a different flag, and we'll get to those next. I turn just the file that Kathy sends me into Excel. I will take the larger of these 2 errors. The units are also different. I need to multiply this times 1,000, whichever is the larger, multiply times 1,000 to get in the normal per mil units for DIC. You can tell I throw away a lot of things because it's repeated. This is the same number all the way down. Same one here. This is, they've got a huge data set that has all their analyses in it. So anyway, I don't need these numbers. I know what they are. I'll reduce the C14 error to a single number, and I don't use the OS number. So when I finish with that winnowing of the data, I wind up with a simple comma separated file that looks like this. And the only thing I've added is I've initialized the flag. So this is station cast bottle. Those are exactly the same numbers as from the previous file, the C14 result. The C14 error, that is the maximum with that internal and external error multiplied times 1,000. C14 flag, I initialize it to 2. We'll get to what the flag values are next. The C13 and the C13 flag, which is also initialized to a value of 2, except for, and do I have an example here? Yes, here's that missing value, the one that didn't have any data. So if the value is missing, it gets initialized with what I use internally as a missing value, which is -999, and it gets a flag value of 5. Flag value of 5 means result not reported, and it doesn't say why. All right, here are the summary of the WHOI flags. We made these up a long time ago, and I'll be the first. I was on the committee that made them, and I'll be the first to admit that we made some mistakes making these flags up, but there are literally thousands of cruises available now that have used these same flags, so it's useful to know what they are. Flag value of 1 is very frequently not even used. If it's in a data set, it simply means that a sample was collected and there is no result in the data set yet, but 1 would be expected. 2 is a good result, 3 questionable, 4 is a bad result, 6 is a result from replicate analysis, typically an average of some sort, and 9 means no sample is collected. There are 7 and 8 flags. Oh, I left 5 out. Okay, this should have a 5 in here. That's the one we had previously, which means the result is not reported. If you like to use ODV for your work, that's the one major program that's floating around the oceanographic community, used for a lot of graphing things. It has a different set of flags, but software exists to do the conversion from the WHOI flags to the ODV flags. As a matter of fact, I think it's Rainer Schlitzer's code. I'm pretty sure it recognizes WHOI flags and automatically does the conversion, but it's a trivial problem at any rate. Okay, what do we do when we find replicates? Now, in this particular cruise, I think there were 543 samples. As you can imagine, scanning through that, looking for replicates would be a tedious and very error-prone job. And some of ours, occasionally we'll get a cruise that might have as many as 1,100 samples in it. So that would really be just unwieldy. There are fairly easy ways to find replicates, but anyway, the first thing we have, what I do in order to find replicates is I generate a unique ID for each sample. And that ID is made up of these first 3 numbers, station, cast, and bottle. If the bottle numbers are small integers, typically 1 through 36, then you can just take the station times 1,000, the cast times 100, plus the bottle number, and you will get a number that's absolutely guaranteed unique. It's by far the simplest way to do it. And I really strongly recommend that. There are all sorts of funny things that people do, having sequential casts, which really confuses things. And at any rate, you need to find some way to come up with a unique ID, and it's got to be guaranteed unique for every cruise, every sample on every cruise. Then in order to find replicates, all you have to do is search the IDs and find the ones that are unique. Most of the high-level programs that people use are MATLAB, Python. I use S+, which is the parent of R. literally has a function in it called unique, and it will find the ones that are unique. And if you do a not on that function, then it finds that will automatically spit out all the replicates. Once you find them, it's quite easy to write a program to average them. I wrote a program like that and then almost immediately threw it away. I much prefer, given that these samples cost, by the time you go out and collect them, get them back to the lab and measure them, they probably cost about $500 a number. My time is fairly expensive, but it's nothing like $500 a sample expensive. So rather than run all of the numbers through an averaging routine, I look at each set of replicates individually, and I will make a subjective judgment. Sometimes there's a number that just should not be included in the average, and so it's rare, but on rare occasions, I will throw a number out. For the average, instead of using the simple average, I use an error-weighted average. For the error estimate, and this here, I mix apples and oranges, and I was well aware of that when I started this procedure. The philosophy behind this is not to be statistically defensible as much as it is to never underestimate the error any more than sort of what we're forced to do. We know we're going to underestimate the errors anyway, but I don't want to make that error, the underestimation error, worse. So I use the larger of the standard deviation of the replicates or the error weighted standard deviation of the mean. Now, given what the errors are, 90% of the time, the standard deviation is the larger number. But occasionally it can come out to be this other one. Either way, I take the larger of those two. For our C13, we use a simple average. And the WHOI standard for C13 data is to not report an error with each data point. Consequently, what we're forced to do is somewhere in the metadata, and there's actually two places to do it, we report an estimated C13 uncertainty for the entire data set. The two places where this shows up, it should always be in the cruise report and the little write-up you do for your cruise report. And the other way to do it is, nowadays we're trying to get people to put what I call a README at the top of every cruise database, at the top of every cruise file. And you can, it's just a really, really terse bit of text. And under the entry up there that it says that C13 was measured and so was responsible. That's a good place to put the C13 estimate for the data set. So then you would have it in two places. This is some of the code that actually does what I've been talking about. I have a little function called ID that I can pass it a cruise file, and it will make the IDs for every one of the samples in that cruise. And hopefully this is big enough on your screens to see. It's I then create a similar set of IDs for the C14, C13 file. And here you can tell on this particular cruise, I just took the station number times 1,000 plus the cast times 100 plus the Niskin number. Then the fun part is hopefully the software system you're using has a function called match, or it won't be called that, but something that works like this. And what match does is it finds it will look at the first value in this junk ID, and it will find it in ID, and it will give the row number where it exists. Then it goes to the second junk ID and so forth. So when I run this generate OK here, OK is going to be an array of locations in this array where these individuals exist. So that's really the hard part of this, is you may have to write your own match function, but hopefully it'll exist in whatever software you use. I do not know of one in Fortran, but they probably exist. Once I generate this array of matchup IDs, then I can run range on it. Now, if there are any that do not exist, then the value of this range function will be NA, and I know I've got to go back and start looking at the data for typos. They are frequent. They can be in either the cruise file or in the data file that comes from NOSAMS. And usually it comes down to a bottle number that got swapped out somewhere on the cruise or something like that. And it'll show up a dozen times the same error. But anyway, this is finds mismatches. And then to find my average, the numbers that I need to average, I can just look for the duplicated. So this duplicated OK is the sort of the inverse of unique. It will find all of the IDs that are duplicated. And so for this cruise, these are the ones that we had duplicates on. So this would be Station 19, Cast 1, Bottle 3, Station 29, Cast 1, Bottle 12, and so forth on down. So once I have this list, I can go in and I start, and here's where the manual part comes. And you might not want to do this, but like I said, I prefer looking at the numbers individually. So this little function AVERROR simply runs the average and standard deviation, just the simple way that we've all been doing that since grade school. Error mean does the error weighted average and standard deviation of the mean, and it spits those out. So I would, for the value for the C14, I'll always use this number, the error weighted average. And for the error, then I would take the larger of the error weighted standard deviation and the regular standard deviation. And this is fairly typical. You can see it's a much larger standard deviation, it's a much larger number. So I would enter those back into the data set, that comma separated file that I showed earlier. Then I just continue on down through the data. That's the most labor intensive part of it. And it can take me an hour and a half to do a big cruise to get through it. And this shows you, this is another representation of that comma separate, exactly that comma separated file that I showed earlier, except we're looking at station 29 because it had a bunch of replicates in it. And this would be what I started with after the averaging. Then I wind up with the modified file that's on the right. So you can see it's just, I do cut and paste to do that rather than use the program I wrote that would do this automatically. Now this part is sort of optional if you're generating a lot of samples the way that we have over the years. Then well, let me back up just a bit. I could take this entire file and submit it to the various data centers and trust them to then merge it into the cruise file. That is what is frequently done in Europe for Pangea. We never got, well, it can happen here, but with the data centers, they're so backed up that if you do that, it may take a year for your samples to get merged into the data file. Rather than do that, I like to merge it myself. Find it's much quicker also. Well, all the errors are on me then if any are made. So that's what this code does. Basically, I read the cruise file in, then I read in that modified data file for the C13 14. You typically have to generate a new column of values because the C13 and C14 won't exist in the data file beforehand. Initialize the values to NA, which in my system is the missing value flag in S+. I initialize the flags to 9, and that's in here somewhere. Here's down at the bottom. Well, let's see. Yeah, in this particular file, the C14 flags had already existed because we gave preliminary version away to a student who wanted to use the data for something. I don't remember what it was. But here I'm going through the same sort of thing where basically I match up the final C14, C13 data file, the ID numbers to those that exist in the entire cruise file, which are these up here, these in what I've called junk. And then I run the match function again. And then once I've generated all of the needed columns and pasted those onto the side of the data file that existed beforehand, then the last thing to do is to fill in the values. And this is where that OK that I generated earlier, this is the array of matchup one file to the other. And then with a simple single statement like this, I can take all 1,000 or how many ever C14 and C13 flags, errors, and values and paste them back into the cruise file that's got everything else going for it. You need something like this rather than to try this would be a major pain if you try to do it in Excel. Largely because in order to see what you were doing, you'd have to hide so many columns. The cruise files now are getting to where they may be 50 columns wide, and that can be unwieldy even on a big screen. So having something like this that is absolutely guaranteed to work is nice. After doing the merge, I go through a QC step, and they're typically two parts to it. The first part is just to examine the data. So I will, once it's imported in, and I keep extraordinarily detailed notes. In this case, this shows one example where for P18, I'm running my QC program and I'm making a plot of pressure versus C14. And this little routine that I wrote allows me to subset the data based on any parameter that exists in the database. So I will virtually always subset by station. I'll only look at a range or a list of stations. And generally the second or third time through, I would also cut on the flag so that I'm not, every time I go through, I don't have to look at the bad data. Because if you get a really bad number, it, of course, it can distort all your scales and everything. But here are the notes I kept for this entire cruise, and I only found one sample that I found 3 that were that I thought were a little bit anomalous, and I made these notes. The translation of these that's the sample ID, and I thought it was probably low versus pressure. One sample was significantly off, and when I put a note like Mark 3, that's my note that says I changed the flag on that one. But at any rate, I will run this sort of code 2 or 3 times, changing the parameter that I'm plotting against. So for C14, good plotting parameters are silicate and alkalinity, if you happen to have those two. And I will continue to edit and add on to these notes. And I prefer to have, I would prefer to not mark a sample unless it differs in at least 2 dimensions. So, if I had high versus, the P is missing here. These are all hand typed, but I would say high versus P and alk and silicate. The other thing that going through this, when any sample that I flag, so here's an example of some of these data. And I found one point that you cannot see, but it's underneath that blue one. I click on it and it comes up and it tells me that station 47 cast 1, and in this case, this is the real bottle number, not the sample, not the position number. At any rate, when I say I've selected all the samples that I want to, then all of the results for this particular sample will show up on my screen, and I can use the results from every parameter that's been measured to influence my decision regarding whether a sample should be marked you should be flagged questionable or bad. So in this case, here's the plots versus it's C14. Oh, and sorry about putting these sideways. I like to, when I'm doing QC, I like to have the independent and dependent variable the way they should be. Anyway, this is a plot of C14 versus pressure. I did it again. Here it is versus alkalinity. Again, another sample that's marked. Here's that same sample. So this particular one, 29.1.1114, is a little bit anomalous versus alkalinity, but when we get to silicate, it's not the least bit anomalous. So the odds are very, very high that I would not mark that point as being questionable. And this is why you use more than one, what I call dimension, when you're looking at this QC. It's very useful to have things that tend to strongly covary. Once I finished making all those plots and keeping all my notes, and you can never ever keep too many notes, then I will make plots of interest. In the cases nowadays, since we're reoccupying lines that have previously been occupied, I will overlay data from the same region the last time it was occupied. And of course, depends on where you are. If you're anywhere in the Atlantic Ocean, particularly in the North Atlantic, I expect to see significant, measurable differences from the top all the way to the bottom. There might be some place in the east of the Mid-Atlantic ridge where that's not true, but I start with the assumption that in the Atlantic we should be seeing C14 differences when the time is 10 years apart, pretty much anywhere. In the North Pacific, that's not true. In the North Pacific, we have not yet seen bomb contamination in the middle of the water column, and in some places in the abyssal values. But at any rate, I'll make property plots similar to those that we saw in the last 2 or 3 plots. I'll also make sections and compare the sections or subtract the sections. And of course, subtracting them gives me a real quick and dirty estimate of change since the last time it was occupied. Also, if, for instance, that data set that Heather mentioned earlier, where there is a bias offset in a data set. So if one lab measured it and another lab, if 2 labs measure the similar set of samples, one really quick way to look at that, you can make property plots, and of course you can see it. Another way to do it that does a bit of averaging and smoothing, so you'll get a better estimate of the cruise difference, would be to make 2 sections and grid 2 sections the same way, and then simply subtract the sections, and you can get an average, actually quite accurate average of the differences that way. At any rate, once I'm finished with the samples, I'm happy with them. I've got all of the values flagged the way that I want them to be. Then comes the part that everyone needs to do every time, and that is to get your data into a database at the quickest possible minute. Now, ever since we started, so going all the way back to CGC 91, the first cruise we did AMS on, we adopted a principle of getting the data into the public realm as quickly as possible. So there have been instances where once the results got to me, it might have taken a month for me to get it out because I had to write some software or something. But as a rule, we have never claimed any proprietary time, and have gotten basically you as soon as I'm finished with the data, it goes to CCHDO and to OCADS, which is the new name for what we used to call CDAC, which was the Carbon Dioxide Information and Analysis System. Basically, this is well, you can see it there. It's that's the data facility out at Scripps. They are very happy to take cruises that, well, I guess I've sent them so many that they will take cruises that are not US cruises. The same thing with OCAD. So these are the two US centers. If you're in Europe, Pangea, your own home country, is going to want to have, certainly have a copy of the data, and they may well want to be the primary repository. That's not an issue anymore. Either one of these places can refer to data that you put into your own national archives by simply linking to it. But the main thing is to get it turned in with the rest of the cruise data and make it publicly available. Now, if you're a graduate student, then all the rules are different, of course. I'm a strong believer in protecting graduate students. So the submission rules are obviously different when students are involved, but do try to get the data in, and remember that it gets to be much harder once your funding has run out for any given cruise. So the most important things I think for this, keep really good records of what you do, get your data into the data archives as quickly as possible. And I have no idea where I am on time, but that's the end of this. Oh, yes.
That was great.
Yeah, these were the other outside notes that I kept regarding the C14, C13 for P18, the P18 cruise. So you can see in January, I got a preliminary version of the data that I merged. The student's name was Bethany Kolodny, who I don't know, but she was doing some sort of student research project, and we had no problem giving her a preliminary version of the data. So WHOI provided the preliminary numbers to me in a file with that name. And I ran them through, merged them, and then sent a copy to Beth. It was 6 months later, roughly in July, I got the final data from WHOI with that name from Kathy. This was the file and the letter that we looked at the beginning of this talk, and I showed the flag changes that, I didn't mention that. Once I finished doing my QC, I always, of course, go back and compare my flags to those suggested by the lab and make sure that we agree. Then let's see, what else did we do here? Yeah, so they sent the data on the 15th, but on the 27th, so it took me 2 weeks, whatever that is. I got around to working on the file, and on that same day, I finished my work, and these notes say that I sent a copy to CCHDO, OCADS, and McNichol. This is Ellen Druffel, because she wanted to use our inorganic numbers to compare to her organic numbers. Kathy at NOSAMS, or Roberta at NOSAMS, and Bill Jenkins was, I don't remember why Bill was involved in this one. For some reason, Bill Jenkins was involved in this one. But at any rate, keeping notes with this level of detail, or even more, well, you'll never regret it. And I've got these notes going back to about 1990. So they're worth copying over and keeping up with, because every once in a while, Steve Diggs, who I note's on the call this morning.
In the chat for you, Bob, about the databases. Steve did.
Beg your pardon?
He put a comment. Steve Diggs put a comment in the chat while you were talking about the databases.
Okay. Yeah, every once in a while, Steve or one of someone at his group will have a question that goes back to something that I did 10 or 15 years ago. And that's when you really appreciate having detailed notes. And what we were seeing on these earlier slides, not those, but these are the exact keystrokes. So it's a keystroke copy with all of the typos filtered out. So I know exactly what I did. And occasionally I can go back and, we'll get a weird result for some reason or the other. This happened not too long ago. And I could go back to the code that I executed and actually found it's it's an issue with S plus that shouldn't exist, but it does. Having, it's just a funny error that popped up. And if you can reproduce exactly what you did, it can be really valuable. Questions?
Yeah, thanks for that, Bob. That's a good lesson in general about data reproducibility. But I just wanted for the group, if you have something for Bob, we wanted to kind of look, we're looking this a bit backwards, but we wanted to give an overview of where ultimately this data ends up, and why it needs to meet certain quality standards. And then in the afternoon or after our break, we'll have the discussions about at the laboratory level, what can we do so that the data can be in a form and have the correct information to go into these databases as Bob just showed. But we have a few minutes. If anyone has any questions for Bob, we can talk about things at this level. And we'll just shift things a little bit.
Yeah, and I apologize for getting down into the weeds with this, but a few of the details are actually have some value.
Yeah, thanks, Bob. This is Rick Wanenkoff. I appreciated the overview, and I know that you always like to be in the weeds. That's where I see you most of the time. So, but this is more of a well, it's more of a comment than a question. One of the things that I've noticed is that it's often very hard to get the actual duplicate info. The individual samples, say when you get to CCHDO, they are basically merged into and get a QC flag of a 6. I would advocate for some sort of means that this duplicate information becomes sort of in the public, easily accessible in the public record, that it's somewhere sort of tied either in the metadata or somewhere else, because there's a lot of useful information that you can get out of replicates, samples, which you don't see if you just do a straight or weighted average.
Yeah, I would not disagree with that a bit, and I would suggest that probably the only, well, there's two ways you could do it. One would get to be unwieldy in a hurry, and the other I think is totally appropriate, and that is in the final cruise report is to include a table of those replicates, and we may have done that back during WHOS. It is not done anymore, but it could certainly be added with no problem. Just to put a table of replicates in the cruise report, because there's no page limits on those, and they are available. They're one click away. If you can get to CCHDO, you can get the cruise report.
Yeah, I think that is, just to interject there, that is, I think is a good idea. That is, as you point out, has been done, quite a bit in the past. But in the more recent cruise reports, you don't see that. But when you have sort of what we call in GOSHIP terms of delayed mode data, like the C13, C14 analyzed on land, and the helium isotopes and others, it often does not get into the cruise report, because the cruise reports mostly are finished, well before the shore-based analysis are done.
Oh, that's true, but we got to get you in the 21st century, Rick. They're saved as they save the files electronically. So I know for a fact that cruise reports are altered years after the final cruise report is turned in. And again, CCHDO does a brilliant job of that. Actually, Steve, if you're on, remind me who out there does that.
Does what part, Bob?
Yeah, who updates cruise reports after the fact?
Oh, that's Jerry Kappa.
Yeah, Jerry Kappa. So if I sent in an addition to the C13, C14 section that included the references now for cruises that occurred 5 years ago, I'm sure that Jerry would put it in the electronic version. And now the only question, again, you get into the weeds, whether or not the DOI for that cruise report would change, I don't know. That's why Alex and the people at CCHDO are always paying attention to the DOI rules and so forth, because we want to have documents that are as accurate as possible. And doing that, you run into technical difficulties with the DOI rules and so forth. It doesn't mean they can't be fixed. It's just, fortunately, they worry about them and I don't have to.
Thanks for that, Bob. We're coming up on a scheduled half-hour break. I would propose we keep the schedule, and then if we come back together as a group for just a few minutes in half an hour, just to talk about what we'd like to talk about, if that is not too redundant in the breakout sessions. It'll be brief, and then we can break out and have our second session discussions, if that is amenable to everyone.
Sounds good.
All right, I don't want to do time zone conversions, but we'll reconvene at noon East Coast.